Hello and welcome to my presentation on a review of the physiology and treatment applications for chronic pain to transcranial direct stimulation. What is transcranial direct stimulation, or tDCS? It involves passing direct current through the scalp, creating a bipolar electrical field to sections of the brain. Two electrodes are used, an anode and a cathode, both placed on the scalp, delivering a direct electrical current, usually 2 milliamperes, creating a bipolar electrical field. This results with the cathode causing a hyperpolarization of the neurons and reduced excitability and an inhibition for action potential. The anode results in depolarization of the neurons, increased excitability of the neurons for an action potential. This is an illustration of the process with blue, the anode, showing the soma of the neuron becoming depolarized, red the cathode, the soma of the neuron becoming hyperpolarized. Although a complete understanding of the physiology of tDCS treatment for chronic pain is unclear, as stated with previous slide, anodal tDCS increases the excitability of the neurons in underlying cortex of the brain, and cathodal tDCS decreases the excitability of the neurons when applied over the primary motor cortex of the brain. tDCS can induce long-term changes at the synaptic level through mechanisms that resemble long-term potentiation for anodal and depression for cathodal stimulation, leading to enhancement of neuroplasticity. It is also suggested that the calcium-dependent synaptic plasticity of glutamatergic neurons is underlying the neuroplastic mechanism of action of tDCS. In addition, biochemical changes have been described, including the number and kinetics of ion channels that could affect the electrical activity propagation and contribute to the non-synaptic plasticity. Brain-derived neurotrophic factor, BNDF, have also been described as a result of tDCS stimulation, a known protein correlated with decreased pain. Previous study of 6 meta-analysis studies in 2018 looked at samples from 48 to 572 participants. From this, 5 of the meta-analysis presented statistically significant positive results for pain management. Points to be considered with this, however, was variability with protocols for tDCS, including differences in electrode placement. And most studies, however, did use anodal stimulation over the primary motor cortex of the hemisphere contralateral to the location of the pain. Another study, which involved a multi-site, double-blinded, randomized, placebo-controlled trial of tDCS, showed with 21 patients and 10 daily tDCS sessions, a significantly less pain interference and pain disability response. Again, further considerations of schedules, the targeted cortical regions, and multimodal approaches were further suggested. However, it was positive. Another study with the implications of function after tDCS treatment was shown with 10 patients with improved function for people suffering from treatment-resistant plantar fasciitis heel pain. This is for elderly people with a mean age of 68. The implications for this is for falls prevention in the future. However, it was acknowledged it was a small sample size. There are limitations with study, with some research showing that there is no significant effect of tDCS. One with 12 participants completed in 2021 showed after 3 consecutive days, there was no difference between treatment and those receiving a sham or placebo treatment. Further additional studies suggested, as stated previously, different cortical targets, neuroimaging to demonstrate engagement with desired cortical targets, and also tDCS to be used with other treatments, including CBT. Thanks very much.